Okay, so uh, hello. So today is the 11th live session of this course, Biochemistry, taken by Professor K. Subramaniam. And in these live sessions, as we know, that we discuss some previous year assignment questions and we try to solve these questions. We try to grasp the concept behind these questions, the basic concept behind these questions, and we learn everything related to those concepts so that it's easier for us to solve similar type of other questions. Okay? So today is the 11th live session where we discuss these concepts, and while we discuss the questions, we can also have an interactive session as we move from one question to another. Okay, so let's start with the first question for today. Give me 1 minute. Okay, so the question for today is blank dash is the analog of coenzyme Q and functions as an electron carrier in the chloroplasts. So what do you think should be here? So I wait for 1 or 2 minutes and then we can discuss the question together. Till then you can try to answer the question. Okay, so the answer for this question will be plastoquinone. Plastoquinone is the analog of coenzyme Q and it functions as an electron carrier in the chloroplast. So if we go into the details of this plastoquinone and the energy transfer mechanism of the chloroplast, so we have to start with the eukaryotic cell organelles. As we know that mitochondria is the energy production center of the cell. It's basically the powerhouse of the cell. And excuse me, We know about the RER, the rough endoplasmic reticulum, and the ribosomes, which basically is the protein synthesis factory of the cell. Then we know about the lysosomes, which is basically the defense and digestion system. Anything foreign particle gets engulfed into the phagosome, which then fuses with the lysosome and ultimately gets digested. And we know about the Golgi apparatus, which is quite complex structure. It helps in synthesis, secretion, and storage of substances. Then we have the microfilaments and microtubules, which provide motility of the cell and cell organelles. So if we look at the schematic of the composite eukaryotic cell, okay, if you see this is an animal. This half represents the animal cell. This half represents the plant cell. The major difference being plant cells have a cell wall, which animal cells don't. Okay. And what is the other major difference? There is a huge part of the plant cytosol being taken up by vacuoles, which is not the case in case of animal cell. Also, we have chloroplasts in the plant cell, which we don't have in case of the animal cells. Rest everything is pretty much similar, okay, equivalent. So this is basically what it is all about. We have see the nucleus, we see the mitochondria in the animal cells over here, the mitochondria in the plant cells over here. We see the chloroplast in the plant cells, and so on and so forth. So the mitochondria, if we go into the history, it was first found by Altman and then named bioblast. It was then named mitochondria much later, and basically this person showed for the first time that they are the respiration center of the cell. Then we talk about the endosymbiotic theory, which basically means endo means within and cytosis, cyto is cell. So this basically means the process of cell eating. That is, cells are engulfed but then usually digested as food. So endosymbiosis means cells are engulfed but not digested. Cells live together in a mutually benefiting relationship, or symbiosis. So what is the role of why do we talk about chloroplast and mitochondria, and we talk about the endosymbiotic theory? So basically the endosymbiotic theory states that the mitochondria and chloroplast in the eukaryotic cells were once aerobic bacteria or prokaryotes that were ingested by a large anaerobic bacteria, which is the eukaryote. Okay, so mitochondria and chloroplast have a double membrane. The inner membrane would have initially been the ingested prokaryote, single membrane. Okay, so that is what the endosymbiotic theory tells us. So numerous lines of evidence exist, including that the mitochondria and chloroplasts have their own circular DNA. Prokaryotes have their circular DNA. Mitochondria and chloroplasts have a double membrane. The inner membrane would have initially been the ingested prokaryote, single membrane, and the outer membrane initially would have come from the cell that engulfed it. Then the mitochondria, chloroplasts that have the 70 ribosomes, so on and so forth. Okay. So this is the endosymbiotic theory. So Lynn Margulis' original hypothesis proposed that aerobic bacteria that require oxygen were ingested by anaerobic bacteria that do not grow well in oxygen, and they may have each had a survival advantage as long as they can continue their partnership. So her hypothesis originally proposed that mitochondria are the result of endocytosis of aerobic bacteria, and chloroplasts are the result of endocytosis of photosynthetic bacteria, and in both the cases by large anaerobic bacteria who would not otherwise be able to exist in aerobic environment. So this arrangement became a mutually beneficial relationship for both cells, symbiotic. Okay. So mitochondria is the energy producing organelle of all cells making aerobic respiration, that is the ATP factory of the cell. The energy obtained by catabolism and oxidation of carbohydrates, lipids and proteins is converted to chemical bond energy in the form of ATP by oxidative phosphorylation in mitochondria. Mitochondria have their own genetic information in a small amount in their own DNA, that is the mitochondrial DNA. They are also involved in calcium homeostasis, apoptosis, radial species generation, radical species scavenging, stellar biosynthesis, etc. So mitochondria is 0.5 to 1 micron diameter, 0.2 to 5 micron in length, oval shaped organelles, and cells without nucleus such as bacteria, green algae, and erythrocytes do not really have mitochondria. Now, form, number, and obviously place changes depending on the cells. For example, they are regularly organized between muscle myofibrils, whereas in spermatozoans, they are aligned in the neck and at the end of the tail. So, they are in greater number in metabolically active cells, which makes sense. And if you talk about the mitochondrial anatomy, the mitochondria contain two major membranes. The outer mitochondrial membrane fully surrounds the inner membrane with a small intermembrane space in between, and the inner membrane separates the mitochondria in two spaces, the matrix and the intermembrane space. So both the membranes are made up of phospholipid bilayers. The inner membrane makes folds named cristae in order to enlarge its surface, and the number of cristae is larger in energy producing cells. So in the part of the inner membrane that looks into the matrix, there are stock particles named inner membrane spheres, which are the parts where ATP synthesis takes place basically. Okay. So if you see, this is the part. So the outer membrane contains porins, which are channel proteins that allow free diffusion of small molecules. They are highly permeable and enzymes of lipid biosynthesis. Okay. That is what the outer membrane is all about. The inner membrane is semi-permeable. That is, some molecules pass to the matrix by active transport. The inner mitochondrial membrane is a functional barrier to the passage of small molecules between the cytosol and the matrix and maintains the proton gradient that drives oxidative phosphorylation. So the inner membrane is parallel to the outer membrane but makes folds named cristae. Now cristae have different structures, and cristae are built in order to enlarge the surface of the inner membrane. So mitochondria and cristae size and number are larger in energy consuming cells. There is the number of cristae of heart muscle mitochondria are 3 times that of the liver mitochondria. Now if you talk about cristae, the cristae, cristae were once thought to be broad wavy folds basically. They are now understood to be more like long caverns. This is a 3D reconstruction of slice cut from mitochondria. Okay. Now, if you talk about the inner membrane, so the inner membrane has a protein lipid ratio of this much. It has no cholesterol, cardiolipin, which is a phospholipid special to mitochondria, and structural proteins, electron transport system, and oxidative phosphorylation enzymes, and transport proteins that regulate input-output of ions and ADP. Okay. Okay. So, and we have the structural proteins, electron transport system, and oxidative phosphorylation enzymes of the respiratory chain, and transport proteins that regulate the input-output of ions and ADP. So, what is cardiolipin? A unique phospholipid which is almost exclusively localized in the mitochondrial inner membrane and present in large amounts in the heart muscle. It plays an important role in energy metabolism. It's the stimulatory cofactor of mitochondrial enzymes, and due to its high negative charge, it restricts the permeability of the inner membrane. Okay, so that is all about cardiolipin. Now the matrix is surrounded by the inner membrane. Proteins, enzymes of mitochondrial DNA synthesis are all there. Ribosomes and tRNA and small amount of granules are also there. So what are the events that take place in the mitochondrial matrix? The tricarboxylic acid cycle, Krebs cycle. Then we have the beta oxidation of fatty acids, oxidation of amino acids, and replication of the mitochondrial DNA. Okay. So the 3 processes of ATP production include glycolysis, the tricarboxylic acid cycle, and the oxidative phosphorylation. Okay, That's the three steps of. So in summary, mitochondria, which is the powerhouse of the cell, are unusual organelles in that they are surrounded by double membrane and retain their own small genome, uh, which also kind of makes them partly autonomous. They also divide independently of the cell cycle by simple fission, and mitochondrial division is stimulated by energy demand. So cells with an increased need for energy contain greater number of these organelles than cells with lower energy needs. So the genetic system of mitochondria is pretty complex and yet pretty self-explanatory. The mitochondria contain their own genetic system which is separate and distinct from the nuclear genome of the cell. So they are thought to have evolved from bacteria that developed a symbiotic relationship in which they live within large cells. So that is the endosymbiotic concept of theory. So this hypothesis has recently been substantiated by the results of DNA sequence analysis, which reveals striking similarities between the genomes of mitochondria and of bacteria such as Rickettsia prowazekii. So Rickettsia are intercellular parasites which, like mitochondria, are only able to reproduce within the eukaryotic cells. So consistent with their similar symbiotic lifestyles, genomic DNA sequences of Rickettsia and mitochondria suggest that they share a common ancestor from which the genetic system of present-day mitochondria has evolved. So if you talk about the mitochondrial DNA, Chevremion et al. Reported for the first time the presence of DNA in mitochondria, and people have done sequence analysis of human mitochondria. The first mitochondrial disease was due to a defect in mitochondria, was also defined. Okay. So the DNA of mitochondria is double-stranded helix, such as the nuclear DNA. It's circular and folded 2 to 3 times. Okay, the human mitochondrial DNA actually contains 37 genes encoding only 13 proteins of the electron transport chain. The other 1500 proteins are encoded by the nuclear DNA. Then we talk about guanine and cytosine bases, which are much more than the nuclear DNA. And genetic code is different from the universal code from nuclear DNA. So this is schematic of the mitochondrial genome. So also there is a concept of the inheritance of mitochondrial DNA, where mitochondrial DNA is genetically transmitted only by the mother to the child. This is maternal inheritance. So there are many copies of mitochondrial DNA in which in every mitochondria, and during cell division, these mitochondrial DNA copies are randomly distributed between the two new mitochondria formed. So if the defect is in few copies as a result of the division, the number of defective mitochondria DNA decreases. So the disease is seen only if the amount of defective mitochondrial DNA exceeds the threshold value. So why maternal inheritance? So any sperm mitochondria that might enter the fertilized egg are destroyed by embryos cellular machinery. So it seems that the male must deposit its arms at the gate of the female. So we can say that the variance of all current living humans can be traced back to a common ancestor on the matrilineal side. It was a woman or who lived years ago. Okay. So inheritance of the mitochondrial DNA standard model of maternal inheritance has recently been challenged because low levels of paternal transmission of mitochondrial DNA have also been observed in crosses between mouse species, but not within species. Although further studies show that this paternal mitochondrial DNA was not transmitted to the subsequent generation. Okay, so that is all about inheritance. So the nuclear DNA is inherited from all ancestors, but the mitochondrial DNA is only inherited from a single lineage. Okay. So mitochondrial disease can be due to these following causes: failure in mitochondrial function, mutations in mitochondrial DNA, mutations in the genes of nuclear DNA encoding mitochondrial proteins. Okay, like mutation like the effect of some drugs like doxorubicin, which is used in cancer therapy, could make mitochondrial damage, DNA damage. It can cause infections. Okay. Several metabolic changes are seen in cancer cells. The mitochondria of cancer cells are different from normal cells in size, content, and location. So, according to the data, mutation accumulations is 10 times greater in mitochondrial DNA than in nuclear DNA. And mitochondrial DNA is a promising molecular marker for cancer as well. So, during the aging process, according to mutations in the mitochondrial DNA, oxidative phosphorylation and energy production actually decreases. And any kind of dysfunctions in the respiratory chain process are seen in late onset neurodegenerative diseases such as Parkinson and Alzheimer. and mitochondrial mutations are recorded in these diseases. Okay. Now, if we come to photosynthesis, if we talk more about the question that we had at hand about mitochondria and chloroplast. So we know that photosynthesis basically leads to chemical bond energy, carbohydrates, lipids, and proteins. Okay. So and the energy comes in the form of ATP or adenosine triphosphate. Correct? This is the structure of adenosine triphosphate. So energy is produced through oxidation of nutrients, like big molecules are cleaved to smaller building blocks. There's proteins to amino acids, polysaccharides to glucose, lipids to glycerol and free fatty acids, all of these. And glucose, fatty acids and the carbon moiety of amino acids are then converted to the acetyl group of acetyl CoA through catabolism. An acetyl group is completely oxidized to CO2 and H2O through reactions of the Krebs cycle. Okay, so this is basically glycolysis, pyruvate processing, citric acid cycle, and electron transport and chemiosmosis. Okay, so the electron transport chain establishes proton gradient that is used to produce ATP. So this is basically the schematic to show where everything happens. Glycolysis can happen in the cytosol, whereas Krebs cycle happens inside mitochondria, in the mitochondrial matrix. Okay, and oxidative phosphorylation occurs around here, along across the mitochondrial membrane. So oxidative phosphorylation is the process in which ATP is formed as a result of the transfer of electrons from NADH or FADH2 to O2 by a series of electron carriers. So this process, which takes place in the mitochondria, is a major source of ATP in aerobic organisms. Okay. And in human organisms, the last step of the oxidation of organic molecules takes place in the so-called respiration or electron transfer chain. So ATP is synthesized with the energy released in this process, which is called oxidative phosphorylation. This energy is converted to chemical bond energy in the form of ATP. Like ADP plus inorganic phosphate will give rise to ATP. So oxidative phosphorylation is the culmination of a series of energy transformations that are called cellular respiration or simply respiration in their entirety. And first, carbon fuels are oxidized in the citric acid cycle to yield electrons with high transfer potential. Then this electron motive force is converted into a proton motive force, and finally the proton motive force is converted into phosphoryl transfer potential. Now, if we talk about the respiratory chain or the electron transfer chain, oxidative phosphorylation takes place on respiratory elements located in the mitochondrial inner membrane, and this system is called electron transfer chain or respiratory chain. Okay, so this electron transfer occurs in the inner membrane of the mitochondria. It is now respiratory chain or the electron transfer chain is a system which comprises enzyme complexes involved in a process in which protons and electrons operated by organic molecules are combined to oxygen in order to form water. So in each phase, 2H plus and 2 electrons are separated from the organic molecules and transferred to another hydrogen acceptor. And the last hydrogen acceptor is oxygen. Okay. This is complex 1, complex 2 and complex 4. So enzymes of the electron transport chain, oxidoreduction enzymes. The prosthetic group of the enzyme, the non-protein part which transports the electrons, is a nucleotide. So nicotinamide adenine dinucleotide NAD+, flavin mononucleotide FMN, flavin adenine dinucleotide FAD. So enzymes of the electron transport chain, apart from NAD and flavoproteins FAD and FMN, 3 types of electron carrier molecules take place in the electron transport chain. Also, we can talk about the iron sulfur clusters as the proteins. There is a non-heme prosthetic group, and obviously the coenzyme Q, okay, ubiquinone, and cytochromes like cofactor iron porphyrin ring, cytochrome B, cytochrome C1, cytochrome C, cytochrome A2A3, cytochrome oxidase. So in cytochromes, iron carries only the electrons. So NAD plus and FAD are powerful electron acceptors. Electrons produced by oxidation reactions are captured by these two: nicotinamide adenine dinucleotide NAD plus, flavin adenine dinucleotide FAD. So NADH and FADH2 carry electron pairs having high transfer potential. They are energy-rich molecules. So they are the cofactors of NAD plus and FAD dehydrogenase enzymes. So the structure of pyridine nicotinic acid, niacin, or nicotinamide, niacinamide. Okay. So nicotinamide adenine dinucleotide is a hydrogen, it's a proton carrier synthesized from niacin. It is basically vitamin B3. So in the NAD plus molecule, nicotinamide is the part responsible of electron transport. Now NAD plus is converted to its reduced form NADH by taking H plus from the substrate. The other proton is released into the medium. Okay, like this. Then we talk about flavin mononucleotide and flavin adenine dinucleotide. So it is a H plus carrier synthesized from riboflavin, vitamin B2. So in FMN and FAD molecules, the isoalloxazine ring is a part responsible of electron transport. This ring transports two electrons and two protons, and the reduced form is FMNH2 or FADH2. So this is the structure of FMNH, FMN, FMNH2. So now what is the role of NADHQ oxidoreductase or NADH dehydrogenase? Its cofactor is basically FMN, flavin mononucleotide. So it contains also dimeric 2 subunits and tetrameric 4 subunits, iron sulfur centers. So iron sulfur proteins have iron sulfur centers containing the iron atoms. Each iron atom is surrounded by 4 sulfurs. So protons and electrons coming from NADH are transferred to electron transport chain by the NADH dehydrogenase enzyme. Protons and electrons are first transferred to FMN to form FMNH2. Okay? And they are then transferred to coenzyme Q via iron sulfur centers. Okay? So this is basically what I have been talking about, the basic schematic of the transfer mechanism. Now, what do iron sulfur centers do? They coordinate a non-heme iron atoms. So, they are prosthetic groups containing 2 to 3, 4 or 8 iron atoms complex to elemental and cysteine sulfur. So, 4 iron centers have tetrahedral structure with iron and sulfur atoms alternating as vertices of the cube. And cysteine residues provide S ligands to the iron, which while also holding these prosthetic groups in place within the protein. Okay, 4 iron sulfur, this is 2 iron sulfur, okay, iron sulfur centers. NAD plus enzymes transfer hydrogens to FMN. So FAD enzymes do not take hydrogens from NAD plus but directly from the hydrogen donor organic molecule. Now we talk about coenzyme Q or ubiquinone. Okay, so this is basically 2,3-dimethoxy 5-methyl-benzoquinone with the polyisoprene side chain on carbon atom number 6. Lipid soluble molecules diffusible in the membrane accepts 2 electrons to become ubiquinol. Okay, this is basic schematic from coenzyme Q and finally to how QH2. So Q transfers only the electrons to electron transport chain, the protons stay in the medium. Cytochrome system or the cofactor iron porphyrin group, sometimes iron or copper ions, sometimes iron and copper ions are together as cofactors. So Fe3 plus Cu2 plus electron acceptor and Fe2 plus and Cu plus can be electron donor. Now what are cytochromes? What are their roles? They are electron transfer proteins with heme ring as prosthetic group. The electron carrier is the iron atom which takes place in the middle of the heme ring, and the iron in heme changes between 2 plus 3 plus valences as during the transfer of the electrons. So heme groups are classified as A, B and C due to their apoprotein structures and differences in their optical densities. Cytochrome B, C1a and H3 are integral proteins of the membrane, whereas cytochrome C is the peripheral protein of the membrane, that is, it resides in the intermembrane space. So each cytochrome has a different reduction potential or a different electron acceptability. So B-type cytochromes have iron porphyrin, protoporphyrin 9 ring. C-type cytochromes have heme C which contains cysteine bridges. A-type cytochromes have heme A which contains isoprene side chain. This is the physical structures of the heme groups. This is cytochrome C. Cytochrome oxidase transfers electrons to oxygen. At the end of the electron transport chain, half mole oxygen is reduced by electrons transferred by cytochrome oxidase and forms water by combining with the proton in the medium. So if we talk about the complexes of the electron transport chain, we have the four enzyme complexes attached with two mobile electron transporters. Three of them, complexes 1, 3 and 4, act as proton pumps. So the four different electron transport complexes are obtained by treatment of the inner mitochondrial membranes with detergents. So the four complexes are NADH-Q oxidoreductase, NADH dehydrogenase complex 1, succinate Q reductase dehydrogenase complex 2, Q cytochrome C oxidoreductase complex 3, cytochrome oxidase complex 4. So there are two mobile electron transporters are coenzyme Q, ubiquitin and cytochrome C. So this is composed of these four enzyme complexes. Okay, the dehydrogenase, coenzyme Q, cytochrome C oxidoreductase, and cytochrome C, hydrochrome C oxidase. So complex 1 is this one. Complex 2 will be the succinate dehydrogenase, succinate Q reductase. Complex 3 will be cytochrome C oxidoreductase, and complex 4 will be cytochrome C oxidase. So this is what happens in the intermembrane and the space of the membranes. Intermembrane space in the matrix. So, what are the prosthetic groups of these enzyme complex protein complexes? NADH dehydrogenase is FMN or FES. Succinate dehydrogenase also FAD or FES. Ubiquinol cytochrome C oxidoreductase heme and FES. Cytochrome C is also heme. Cytochrome oxidase is heme CU and CUB. So enzymes with FAD as prosthetic group transfer the electrons directly to Q. Contrarily to NADH-Q oxidoreductase, they do not pump protons to the cytosolic side of the inner mitochondrial membrane. So this is why the number of ATP molecules produced by the transport of electrons to FADH is less than the number of ATP molecules produced by the transfer of electrons to NADH. So NADH dehydrogenase transfers hydrate ion from NADH to ubiquinol and pumps for protons across the inner mitochondrial membrane. So these protons are pumped out of the mitochondrial matrix. Therefore, a proton gradient has been built up, and the matrix becomes negatively charged, and the intermembrane space becomes positively charged. Complex 1 is L-shaped. The peripheral domain containing the FMN that accepts 2 electrons from NADH protrudes the mitochondrial matrix. FES centers are also located in the hydrophilic peripheral domain where they form a pathway for electron transfer from FMN to coenzyme Q. Electrons pass through a series of FES centers and are eventually transferred to coenzyme Q. Succinate dehydrogenase of the Krebs cycle is also called succinate CoQ reductase. So succinate is oxidized to fumarate and FAD is reduced to FADH2, and FADH2 transfers electrons through coenzyme Q to the respiratory chain. So at the end of the Q cycle, two QH2 oxidized and 2Q are produced. Two cytochrome of C molecules are reduced. One cytochrome C molecule can carry only one electron. Two cytochrome C must react to carry the 2 electrons of QH2. The 4 protons, 2 from the matrix and 2 from Q, are pumped across the inner mitochondrial membrane to the inner membrane space. What is complex 3? The ubiquinol cytochrome C oxidoreductase transfers electrons from ubiquinol to cytochrome C. Cytochrome oxidase carries out the following reversible reactions. The 4 electrons are transferred into the complex one at a time from cytochrome C. So this is basically the schematic that covers the entire electron transport chain. Okay. So where is the ATP synthesized? It's in the head part of the mitochondrial inner membrane spheres. ADP plus PI is ADP. So F0, F0, F1 ATPase or ATP synthesis of complex V. Complex 5. This is the stalk. This portion is the stalk and this portion is the head. So ATP synthesis is in the enzyme complex embedded in the inner membrane of mitochondria. It seems like a sphere, F1 on a stalk. So this is the stalk and this is So the F1, F0 ATPase, the inner membrane spheres constitute the ATP synthase enzyme itself. So ATP synthase can be separated into two subunits. F0 stop is a proton channel embedded in the inner membrane. When protons flow through this subunit, rotation occurs and production of ATP from ADP and PI is catalyzed. But the F1 head is spherical portion projected into the matrix of the mitochondria, the place where ATP synthesis takes place. Okay. So how is ATP synthesized? The protons released by the transport of electrons to the enzyme complexes are pumped from the matrix to the intermembrane space. This pumping produces a pH gradient and a transmembrane electric potential. So the motive force provided by these factors provokes a pumping of protons back from the intermembrane space to the mitochondrial matrix, passing through F1, the ion channel portion of the ATP synthase. And this proton motive forces give rise to the coupling of ATP by ADP and PI in the spherical portion if not of ADP synthase. Okay, so the protons released by the transport of electrons to the enzyme complexes are pumped from the matrix to the intermembrane space, and this pumping produces a pH gradient and a transmembrane electric potential. So the motive force provided by these factors provokes the pumping of protons back from the intermembrane space to the mitochondrial matrix, passing through F1, the ion channel portion of ATP synthase. So this proton motive forces gives rise to the coupling of ATP by ADP and PI in the spherical portion of ATP synthase. Okay, so if we talk about the chemiosmotic hypothesis, it was proposed by Peter Mitchell, and this hypothesis stated that a proton motive force was responsible for driving the synthesis of ATP, and in this hypothesis, protons would be pumped across the inner mitochondrial membrane as electrons went through the electron transfer chain, and this would result in a proton gradient with a lower pH in the intermembrane space and an elevated pH in the matrix of the mitochondria. So an intact inner mitochondrial membrane impermeable to protons is a requirement of such a model. So the proton gradient and the membrane potential are the proton motive force that is used to drive ATP synthesis. So in effect, the pH gradient acts as a battery which stores energy to produce ATP. So over the past several years, Mitchell's chemiosmotic hypothesis has been widely accepted as the mechanism of coupling of electron transport and ATP synthesis, and he was awarded the Nobel Prize in Chemistry in 1978. And this acceptance by the scientific community is a result of accumulating experimental evidence supporting the hypothesis. Okay, so this is the chemiosmotic hypothesis that protons are pumped across this membrane as electrons flow through the respiratory chain. So the pH outside is 1.4 units lower than inside. The outer mitochondrial membrane, the inner mitochondrial membrane, and intermembrane space. So membrane potential is this. So electron transport through the respiratory chain leads to pumping of protons from the matrix to cytosolic side of the inner mitochondrial membrane. The pH gradient and the membrane potential constitute a proton motive force that is used to drive ATP synthesis. So this is ATP synthase. Proton pumps of the respiratory chain. Protons pumped to the intermembrane space to electron transfer and pumped back to the matrix, passing through the ATP synthase rotor. And ATP synthesis is repowered by the potential energy stored in the gradient of protons. So the flow of ions through a membrane is called chemiosmosis. The flow of protons through the mitochondrial membrane is chemiosmosis. ATP synthase. Uh, ATP synthase. This synthesizes ATP from ADP and inorganic phosphate. So ATP synthase is like a rotor driven by protons. Okay. So the backflow of protons to the inner membrane of the mitochondria back to the matrix basically provides the power that generates ATP. So like the flow of water flips the water mill, the flow of protons turns the ATP synthase wheel. Basically the same concept. So in summary, for the synthesis of one ATP, the number of protons that should pass from the cytosol to the matrix is approximately 3. One proton is used for the passage of one ATP after being synthesized by ATP synthase and from the matrix back to the cytosol in order to be used in the metabolic reactions. Total of 4 hydrogens or protons needed for the synthesis of one ATP. So in the case of the oxidation of one NADH, the 10 protons pumped to the cytosolic side of the membrane could achieve the synthesis of 2 to 5 ATP molecules. Then in the case of oxidation of one FADH2, the 6 hydrogen protons pumped to the cytosolic side of the membrane could achieve the synthesis of 1,5 ATP molecules. Okay. This is the mitochondrial electron transport chain. The matrix is a high pH and low hydrogen concentration. So these are some of the inhibitors of oxidative phosphorylation and the site of action. Cyanide basically works on complex 4. Amidal works on complex 1. Rotenol on complex 1. Antimycin complex 3. All of these are electron transport inhibitors. Okay. Now, if we start talking about plant cells, plants cells are readily distinguished from animal cells by the presence of two types of membrane-bounded compartments, vacuoles and plastids. So, both the organelles are related to the mobile lifestyle of plant cells. So, plastids are present in all living plant cells and in euglena. They are small bodies found free in cytoplasm. Then the chloroplast is most widely occurring chromoplast of the plants, and it occurs mostly in the green algae and higher plants. So the chloroplast contains the pigment chlorophyll and chlorophyll A and chlorophyll B and DNA and RNA. Okay, so according to Swimfer, different kinds of plastids can transform into one another. The chloroplasts remain distributed homogeneously in the cytoplasm of plant cells, but in certain cells, the chloroplasts become concentrated around the nucleus or just beneath the plasma membrane. The chloroplasts have a definite orientation in the cell cytoplasm. So, since chloroplasts are motile organelles, they show passive and active movements. So, if you talk about the shape, higher plant chloroplasts are generally biconvex or planoconvex. However, in different plant cells, chloroplasts may have various shapes. Okay, the size of the chloroplast varies from species to species, and the chloroplasts of polyploid plant cells are comparatively larger than the chloroplasts of the diploid plant cells. So generally, the chloroplasts of plants grown in the shade are larger and contain more chlorophyll than those plants grown in sunlight. So the number of chloroplasts varies from cell to cell and from species to species and is related with the physiological state of the cell, but it usually remains constant for a particular plant cell. So the algae usually have a single huge chloroplast. Cells of the higher plants have 20 to 40 chloroplasts. So according to a calculation, the leaf of this particular contains about 40,000 chloroplasts per square millimeter of surface area. So when the number of chloroplasts is inadequate, it is increased by division. When excessive, it is reduced by degeneration. So when we talk about envelope, the entire chloroplast is bounded by an envelope which is made up of a double unit membrane. So across this double membrane envelope occurs exchange of molecules between chloroplast and cytosol. And isolated membranes of envelope of chloroplast lack chlorophyll, pigment and cytochromes, but they have a yellow color due to the presence of small amounts of carotenoids. So they contain 1 to 2% of the total protein of the chloroplast. In case of the stroma, the matrix of stroma fills most of the volume of the chloroplast and is a kind of jelly fluid phase that surrounds the thylakoids. So it contains about 50% of the proteins of the chloroplast, most of which are soluble type. So the stroma also contains ribosomes and DNA molecules, like 80 DNA molecules per chloroplast per cell of Chlamydomonas, 20 to 40 DNA molecules per chloroplast per cell of leaf or maize, and both of which are involved in the synthesis of some of the structural proteins of the chloroplast. The stroma is the place where CO2 fixation occurs and where the synthesis of sugars, starch, fatty acids, and some proteins takes place. So the thylakoids consist of flattened and closed vesicles arranged as a membranous network. So the outer surface of the thylakoid is in contact with the stroma and its inner surface encloses an interthylakoid space. So thylakoids may be stacked like a neat pile of coins, like if you have seen the structure forming grana, or they may be unstacked, intergranal or stromal thylakoids forming a system of anastomosing tubules, okay, that are joined in the grana thylakoids. So there may be 40 to 80 grana in the matrix of a chloroplast. So the number of thylakoids per grana may vary from 1 to 50 or even more. Okay. So these are the images of the structure of chloroplasts, thylakoids, granum and stroma. So the molecular organization of the membrane of thylakoids is based on the fluid mosaic model of the membrane, which represents the following characteristics: fluidity, asymmetry, and economy. And lipids represent about 50% of the thylakoid membrane. And these include those directly involved in the photosynthesis, such as chlorophylls, carotenoids, and plastoquinones. Okay, that was all the question was all about. Now, structural lipid thylakoids of lipids of thylakoids include glycolipids, sulfolipids, and a few phospholipids. And most of these structural lipids are highly unsaturated, which confer to the membrane of thylakoids a high degree of fluidity. Okay. So the protein components of thylakoid membrane are represented by 30 to 50 polypeptides, which are disposed in the following 5 major supramolecular complexes. Okay, the photosystem 1, which basically is a complex that contains a reactive center composed of P700. Okay, because it's type of pigment that gets bleached at the wavelength of 700 nanometer. Several polypeptides, a lower chlorophyll A-B ratio, and beta carotene. It acts as a light trap and is present in unstacked thylakoid membranes. In it, light-induced reduction of NADP plus takes place. Then we have photosystem 2. This complex comprises two intrinsic proteins that bind to the reaction center of chlorophyll P680. Because the pigment that bleaches when absorbing light at 680 nanometer, it contains a high ratio of chlorophyll A, B, and beta carotene. Frequently, the PS2s are associated with the light harvesting complex and are involved in light-induced release of O2 from H2O. PS2 works as a light trap in the photosynthesis and is mainly present in the stacked thylakoid membranes of grana. Cytochrome B and F. This complex contains one cytochrome F, two cytochromes of B, 563, one iron sulfur center and a polypeptide. It is uniformly distributed in the grana and acts as the electron carrier. These two complexes are related to the electron transport and are linked by mobile electron carriers, that is plastoquinone, plastocyanine, and ferredoxin. This electron transport through pH2 and pH1 finally results in the reduction of the coenzyme NADP+. Simultaneously, the transfer of protons from the outside of the to the inside of the thylakoid membrane occurs. Then we have the ATP synthetase. It's as in mitochondria. This complex consists of a CF, not hydrophobic portion, a proteolipid that makes the proton channel, and a CF1 that synthesizes ATP from ADP and PI using the proton gradient provided by the electron transport. So ATP synthetase complexes are located in the stack membrane. Light harvesting complex. The main function of light harvesting complex is to capture solar energy. It contains two main polypeptides and both chlorophyll and B. Light harvesting complex is mainly associated with photosystem 2 but may also be associated with photosystem 1. And light harvesting complex is localized in stack membranes and lacks photochemical activity. So what are the functions of the chloroplast? The light reaction is also called Hill reaction, photosynthetic electron transfer reaction, or photochemical reaction. In the light reaction, solar energy trapped in the form of chemical energy of ATP and is reducing power in NADPH. During it, oxygen is evolved by photolysis or splitting of water molecule. Light reaction occurs in thylakoid membranes. The dark reaction is also called Calvin reaction, photosynthetic carbon reduction cycle, carbon fixation reaction, or thermochemical reaction. And in the dark reaction, the reducing capacity of NADPH and the energy of ATP are utilized in the conversion of carbon dioxide to carbohydrate. Such a process of carbon fixation or CO2 fixation occurs in the stroma of chloroplast. So this is a light dependent reaction. This is the Calvin cycle. So the light reaction, the most important step of the light reaction is harvesting of the maximum amount of solar energy for conversion into chemical energy. So the photosynthetic light reaction is completed by passing through the following process, like light absorption by photosynthetic pigment. And it was Einstein who suggested that light and other electromagnetic radiations travel in discrete packets called quantum photons, right? And that when light interacts with matter, it does so by annihilating complete photons, never part of one. So further, according to Einstein's photoelectric theory, it takes one photon to eject one electron. Increasing the density of light or flux of protons will increase the number of electrons ejected, not their velocities. On the other hand, changing the wavelength of light does change the velocity of ejected electrons, implying that the energy of a photon must be related to its wavelength. So when a molecule absorbs a photon of light, it is absorbing a quantum of energy. Several things can happen to this, like it can be dissipated in molecular motion, manifested as heat. It can be re-emitted as a new photon of light at a longer wavelength with a shift representing losses to other processes. If re-emission occurs very quickly, it is called fluorescence. If there is a long lag between absorption and re-emission, the process is called phosphorescence. And the energy of light can cause a chemical change in the compound that absorbs it. So it is the latter possibility that can happen when a molecule of chlorophyll absorbs a photon. So in fact, chloroplast acts as an energy transducer. That is, it can convert light energy to chemical energy, much as a solar battery uses light to run a transistor radio. So within a fraction of a second after light is absorbed by a photosynthetic pigment, the molecule is altered. Some electrons associated with the pigment are raised to new energetic heights, changing their spin or modifying their position. And if enough energy is absorbed, an electron may even be ejected, and thus oxidation will occur. So when these events happen, we say that the molecule is in excited state. Okay. And if we talk about the photosynthetic units, it seems to be a group of roughly 300 pigment molecules located in the chloroplast membrane. There is a thylakoid disc. So although all the pigment molecules in the unit, like carotenoids, chlorophylls, are capable of capturing light energy, they must transfer it to a single key chlorophyll A molecule called the reaction center. So the latter then loses an electron to a series of electron carrier molecules. The other 299 auxiliary pigment molecules are referred to as antenna molecules or antenna pigments to designate their role in the capture of light energy. Higher plants are found to have two types of photosynthetic units associated with two different pigment systems which absorb light of different wavelengths. So pigment system 1 or photosystem 1 units occur in the thylakoid membrane in the form of small and densely packed particles. So each PS1 or photosystem 1 unit consists of about 200 molecules of chlorophyll A and 50 carotene molecules. So the reaction center is called P700 because it has maximum light absorption at 700 nanometers. The energy funneling into P700 is responsible for the ejection of an electron from chlorophyll. Pigment system 2 or photosystem 2 units occur in the thylakoid membrane in the form of larger, more widely spaced particles or AS particles or quantosomes. So each PS2 unit consists of approximately 200 molecules of chlorophyll A, 200 molecules of carotenols, chlorophyll B and chlorophyll C or D depending upon the species. This reaction center chlorophyll A is designated P690, a shorter wavelength trap. So in the thylakoid membrane, PS1 and PS2 are probably arranged near one another, forming the so-called Z scheme, okay, because they are functionally related. So excitation energy originating in one can be shunted to the other system. Okay. However, two processes can uncouple chemically rather than through direct energy transfer. Okay. So electron transport systems and oxidation of water. So when the P680 of photosystem 2 acquires a sufficient quantum of energy, it emits a pair of electrons. These electrons with high potential energy can move down an electron transport chain, and during this process ATP molecules are formed. So two electrons are passed to an electron transport acceptor Q, which is a quinone electron transport chain involving 4 electron carriers plus 2 quinone before being passed on to PS1. So the electrons are passed through 4 carriers successively at lower energy levels so that at each step energy is released, which is harvested in the production of 2 ATP molecules. So the electron lost by P680 is ultimately accepted by P700 of photosystem 1, which also captures light, and for absorbing each proton it ejects an electron. So this electron is replaced by an electron from PS2 or photosystem 2, and this flow of electron continues as long as the light is available. So electrons liberated from P700 are passed to acceptor to an electron transport chain like ferredoxin NADP reductase at successive lower energy levels, and finally these electrons reach NADP coenzyme, each molecule of which receives an electron, enabling it to pick up a proton, thus producing 2 molecules of NADPH from 1 molecule of H2 used in PS2. Okay, that's basic schematic. Okay, and oxygen is given out by these photosynthesizing plants. The protons accumulate inside the thylakoid membranes, resulting in a proton gradient, and the energy released by the protons when they diffuse across thylakoid membrane to the stroma along the proton concentration hydrogen plus concentration gradient is used to produce ATP molecules by this F0 F1 ATP synthetase of the membrane. So as ATP synthesis occurs in light and the process is not cyclic, that is, it needs a constant supply of water molecules to be oxidized and NADP to be reduced. The process is called non-cyclic photophosphorylation. Okay, and ADP production by cyclic photophosphorylation also occurs during the light reaction. So this process involves another electron transfer mechanism involving cytochrome B6 and starting with P700. So the ultimate acceptor of the de-energized electron is also P700 of photosystem 1. So all the molecules of ATP and NADP generated in the light reaction of photosynthesis are used by cellular enzymes and stroma of chloroplasts during the dark reaction. Total light reaction can be summarized like this. Okay. And the dark reaction can be completed through these phases. Phase 1 will be carboxylation, where the 3 molecules of carbon dioxide will be attached to 3 molecules of ribulose 1,5 bisphosphate, okay, to produce short-lived 6 carbon intermediates, and this process is called carboxylation and is catalyzed by the enzyme rubisco, which is the most abundant protein present on planet Earth. And rubisco is a large protein molecule comprising 60% of the chloroplast protein. So these 6 carbon intermediates are individually broken down into 6 molecules of 3-phosphoglyceric acid. Okay. In the phase 2, it's glycolytic reversal. So by utilizing 6 ATP molecules, these 6 molecules of PG are transformed into 6 molecules of 1,3-diphosphoglyceric acid. These in turn get converted into 6 molecules of glyceraldehyde 3-phosphate, 3-phosphoglyceraldehyde or 3-phosphoglyceric acid by utilizing 6 NADPH molecules. And then phase 3, regeneration of RUBP. For the continuous running of the Calvin cycle, there must be a regular supply of ATP and NADPH and also sufficient amount of RUBP. So, 3 molecules of RUBP are regenerated by a complex series of reactions which utilize 3 ATP molecules and 5 molecules of 3-phosphoglyceric acid. This leaves 1 molecule of PGAL as the net gain of 1 Calvin cycle. So 2 turns of Calvin cycle result in the production of small molecule of glucose, and this glucose is used by the plant to form a large variety of organic compounds required for its structure and function. Okay. So this in eukaryotes photosynthesis occurs in chloroplasts. You can see over here. So the major side steps of photosynthesis are over here. Photon absorption excites an electron. Electron flows is plus to permanent region of high concentration positive charge, and H plus flows out of the high concentration positive charge. Okay. So these are the major steps of photosynthesis. If you can see the entire schematic over here. Okay. So the question that we had started with was about plastoquinone, analog of coenzyme Q, which is basically nothing but plastoquinone that we have talked about. Okay. Now we'll move to the question. Let us start with the question that let us go back to the question that we started with. So that's the analog of coenzyme Q and functions electron carried in the chloroplast. The answer will be plastoquinone that we have already discussed in the while we are discussing chloroplast, right? Move on to the next question for today. So this is plastoquinone as a proton pump. So whatever we discussed today, you can think this photosystem to the initial site of excitation. Okay, if you can see, this is basically what happens. It acts as a proton pump. So this clever recycling of electrons in a loop is called the Q cycle. So electron transport chains in photosynthesis and respiration both use these Q cycles. If you can see the plant chloroplast in cyanobacteria and in mitochondria as well. So we talked about all of this. So we'll move on to the next question for today. Give me a minute. So this is the next question for today. Match the following. So we have a number of diseases in one of the columns and a number of deficiency on the other column. So you have to match the diseases with the deficiency of the vitamins. So let me know. So just think about this for some time, then we can discuss this again. Okay, so the answer will be A4, B1, C2 and D3. So we'll discuss about all the vitamins and the diseases one by one. So if we talk about vitamin E describes a family of 8 antioxidants, 4 tocopherols and 4 tocotrienols. So alpha tocopherol is the active form of vitamin E in the human body. Okay, and the 4 tocopherols will be alpha, beta, gamma and delta. So the main function of vitamin E is antioxidant. It intercepts free radicals and prevents destruction of cell membrane. So it protects the fat in LDL, low density lipoprotein, from oxidation. It inhibits platelet aggregation. It enhances vasodilation. It inhibits the activity of protein kinase C. Correct? And what are the dietary sources of vitamin E? Vegetable oils, almonds and peanuts, avocado, spinach, carrots, all of these. So what can vitamin E deficiency cause? So severe vitamin E deficiency can cause neurological symptoms like impaired coordination and muscle weakness, increased risk of cardiovascular diseases, hemolytic anemia in children. The risk factors will be genetic defect affecting the transfer protein of alpha tocopherol. Okay, so what are the therapeutic uses? So it's prevention of cardiovascular disease, diabetes mellitus, cancer prevention, it boosts immunity, dementia, etc. So toxicity, excess vitamin E may cause impaired blood clotting, leading to increased risk of bleeding in some person. So it is recommended that vitamin E supplements to be stopped 1 month before any kind of surgery. For vitamin K, it is derived from the German word coagulation. So there are two naturally occurring forms of vitamin K. Plant synthesize phylloquinone, vitamin K1, and bacteria can synthesize menaquinone 3, vitamin K2. So menaquinone 4 is produced in animals from vitamin K1, but its function is yet to be explored. So vitamin K is needed for production of vitamin K dependent coagulation factors in the liver. Other functions include assisting in bone mineralization. The mineral binding capacity of osteocalcin requires vitamin K. Okay, and gas6 is also a vitamin K dependent protein identified 993. It's important for neuronal function. So what are the sources of vitamin K? So bacteria in large intestine produces vitamin K2 and supply 40 to 50% of human requirement. Okay, like vegetable oils, almonds and peanuts, avocado, broccoli, spinach, lettuce, parsley, etc. So vitamin K deficiency is quite uncommon in adults. Only those with severe liver disease or those on oral anticoagulants are at risk. So exclusively breastfed and premature babies are at risk because human milk is low in vitamin E and their gut is not yet colonized with bacteria. So hemorrhagic disease of the newborn is a serious threat to life, and routine vitamin K prophylaxis is recommended by doctors. So if we come to vitamin C, humans, unlike other mammals, are unable to make ascorbic acid and they get it from food. So there are rich dietary sources: citrus juices, strawberry, guava, tomato, sweet red pepper and broccoli. So the recommended daily intake is between 15 to 120 mg per day depending on age. Smokers and lactating mother needs a higher range. So what are the functions? Collagen synthesis, antioxidant synthesis of noradrenaline, carnitine synthesis and metabolism of cholesterol to bile salts. If we talk about vitamin C deficiency, severe deficiency can lead to scurvy with following manifestations like bleeding and bruising easily, hair and teeth loss, joint pain and swelling, fatigue and lack of concentration. Therapeutic uses can be cardiovascular diseases, cataracts, diabetes mellitus, cancer prevention, common cold, lead toxicity, etc. Now, contraceptive pills and aspirin lower vitamin C level in plasma and WBC. So, vitamin C in large dose blocks the action of warfarin and interferes with interpretation of certain lab tests. Okay, so previous claims of serious toxic effects of vitamin C are not really evidence based. Okay, now if we talk about the vitamin B complex, these are a group of 7 water soluble vitamins: thiamine, riboflavin, niacin, pyridoxine, cobalamin, biotin, and pantothenic acid. So biotin and pantothenic acid deficiencies are extremely rare because it is found in numerous foods and also is synthesized by intestinal bacteria. Now biotin deficiency may occur with prolonged antibiotic therapy and ingestion of raw eggs. Thiamine is rapidly converted to its active form, thiamine pyrophosphate, in the brain and liver by a specific enzymes, thiamine diphosphortransferase. Now, this TPP is necessary as a cofactor for the reactions of the pentose phosphate pathway. So, the dietary requirement for thiamine is proportional to the caloric intake of the diet and ranges from 1 to 1.5 mg per day for normal adults. Now, low intake and alcoholism is a risk of thiamine deficiency, increased consumption, excessive loss of hemodialysis and diuretics, anti-thiamine factors, all of these. Now, what are the deficiencies? Severe thiamine deficiency can lead to beriberi, Wernicke-Korsakoff syndrome. So, thiamine is used for treatment of congestive heart failure and Alzheimer's disease as well as in cancer prevention. Now, riboflavin, vitamin B2. Adequate amounts of B2 is present in eggs, milk, meat, and cereals, and deficiency can often be seen in chronic alcoholics due to their poor dietary habits. Like symptoms associated with riboflavin deficiency include glossitis, seborrhea, angular stomatitis. Riboflavin decomposes when exposed to visible light, so this characteristic can lead to riboflavin deficiencies in newborns treated by phototherapy. Now, what is niacin? Niacin is available in both animal and plant food and is made in the body from tryptophan. So, severe deficiency causes pellagra with glossitis, dermatitis, diarrhea, depression, and dementia. And in large doses, niacin lowers plasma cholesterol, but it elevates blood glucose and uric acid levels. It's recommended with not recommended with diabetes and gout. Then we come to pyridoxine, which functions as a cofactor in enzymatic reactions required for the synthesis and catabolism of the amino acids as well as glycogenolysis. Vitamin B6, it's widely available in diet and deficiency may follow INH and penicillamine therapy. The deficiency can cause neonatal seizures, chelosis, glossolitis, and all of this. Neuritis. Now, B12 functions as a cofactor for enzymes required for the catabolism of fatty acids and the conversion of homocysteine to methionine. So, B12 is not available in plant and deficiency may occur in strict vegetarians and in patients with GIT problems and those on prolonged antibiotic treatment. Okay? And deficiency causes megaloblastic anemia and high homocysteine in blood, which is a risk of stroke. Then we come to folic acid, which is obtained from yeast and leafy vegetables as well as in animal liver. Animals can't synthesize folate, thus it must come from diet. So folate is needed for synthesis of nucleic acids, and deficiency causes megaloblastic anemia and neural tube defects in utero. So it's used for the treatment of chronic hemolytic anemia. Now, vitamins are essential organic nutrients required in small amounts. They cannot be synthesized by the body. So they must be obtained by outside sources like diet, human bacteria, and sun. They are required for growth, maintenance, reproduction, and lactation. So these are the classes of vitamins. Soluble vitamins are stored in tissues. Water soluble vitamins are not stored in tissues, must have constant supply. So what are the function, deficiency, signs, and sources of vitamin A? Function is development of healthy skin and nerve tissue. Aids in building up resistance to infection. Functions in eyesight and bone formation. All animals require a source of vitamin A. It is important in the ratio of pregnant females. Deficiency is retarded growth in the young, the development of a peculiar condition around the eyes known as xerophthalmia, night blindness, and reproductive disorders. Sources can be whole milk, carotene, animal body oils, legume forages, and can be synthetically produced. Then we have vitamin E, which function is normal reproduction. Deficiency signs are poor growth, crazy chick disease, muscular dystrophy. Sources can be synthetic for poultry and swine, cereal grains and wheat germ oil, green forages, protein concentrates, oil seeds, etc. And vitamin E rapidly destroyed in rancid or spoiled fats. That is why these may cause white muscle disease. Utilization of vitamin E is dependent on adequate selenium. Then we come to vitamin D, which is essential for the proper utilization of calcium and phosphorus to produce normal healthy bones. Deficiency will be retarded growth, misshapen bones, lameness, and osteoporosis. Sources can be whole milk, forage crops, irradiated yeast, etc. Then vitamin K, necessary for the maintenance of normal blood coagulation. Deficiency signs can be blood loses its power to clot or the time needed for clotting is longer. And serious hemorrhages can result from slight wounds or bruises. Sources can be green leafy forages, fish meal, liver, soybeans, etc. Vitamin C is ascorbic acid. Function is has an effect on the metabolism of calcium in the body. None demonstrated in livestock. Human deficiency can be scurvy and brittleness of bones. Sources can be citrus fruits, tomatoes, leafy vegetables, etc. Vitamin B1 is thiamine, required for the normal metabolism of carbohydrates, loss of appetite, muscular weakness. Sources can be raw whole grains and especially their seeds and embryos. Now, vitamin B2 is riboflavin. Function is necessary for normal embryo development, important in the metabolism of amino acids and carbohydrates. Deficiency signs will be poor reproduction, categorized by small uterus and deformed young, and sources will be milk, dairy byproducts, yeast, green forages, all of these. So we know about minerals. Minerals are essential inorganic nutrients required in small amounts, required for growth, maintenance, reproduction, and lactation. So these are the macro minerals: calcium, phosphorus, potassium, sulfur, sodium, chlorine, magnesium. Calcium is a major component of bones and teeth and essential in blood coagulation, nerve and muscle function, and milk and egg production. It deficiencies are retarded growth, deformed bones in young animals. Sources will be milk, oyster shells, and limestone. Sodium chloride, they are considered together because of their close biochemical relationship, and they are always provided as common salt. They are required for the formation and retention, concentration, and pH of body fluids such as protoplasm, blood. It's important in the formation of digestive juices and functions in nerves and muscle activity. Deficiency will be poor condition and depressed appetite. Most farm-produced feeds are deficient in these two minerals. Sources are salt supplements and injectable products. Phosphorus will be essential for the formation of bones, teeth, and body fluids. They are required for metabolism, cell respiration, and normal reproduction. Deficiency signs are similar to calcium deficiency. Sources will be dicalcium phosphate, bone meal, and low fluorine phosphates. Potassium retention and formation of body fluids, pH concentration of body fluid, and rumen digestion. Deficiency signs non-specific and unlikely under most conditions. Manganese will be helpful in fetal development, adult development, milk production, and skeleton development. Deficiency will be causing abortion, reduced fertility, deformed young, and poor growth. Sources will be trace mineralized salt. The microminerals are iodine, copper, iron, selenium, manganese, molybdenum, zinc. Copper should be present in animal tissues for iron to be properly utilized, hemoglobin formation and synthesis of keratin for fair and whole growth. Deficiency signs: poor pigmentation of feathers, stingy wool, sources forages and copper salts. Iron will be essential for the function of every organ and tissue of the body. Deficiency will be seldom occurs in older animals. Nutritional anemia. Sources are forages and copper or trace mineralized salts. Cobalt is required as a nutrient for the microorganisms in ruminants and thereby aids in human synthesis of vitamin B12. Okay, deficiency will be lack of appetite, loss of weight, rough hair coat, anemia, decreased milk and wool production. Sources will be legume forages and salt containing cobalt. Magnesium, similar to calcium and phosphorus, animals are irritable, their heartbeat will be irregular. Mineral supplements and ordinary feeds. Okay, so that was all about the vitamins. So we'll go to the next question for today. Yeah. So the next question for today is: the enzyme for phosphorylation of fructose phosphate is called dash. To separate it from another enzyme dash that catalyzes the formation of dash from fructose 6 phosphate in a separate pathway. In some organisms, dash is the potent allostatic activator of dash. These are the options. What do you think is the correct answer? Anybody? Okay, so basically the answer will be PFK2, PFK1, fructose 2,6 phosphate. Okay, so if we go through the process, central role of glucose, let's start with that. So the central role of glucose is one and many. It can give rise to storage, glycogen, starch, sucrose, oxidation by glycolysis to give rise to pyruvate, oxidation by pentose phosphate pathway to give rise to ribose 5-phosphate, and synthesis of structural polymers, extracellular matrix, and cell wall polysaccharides. Okay, so this is overview of glycolysis. The phase one will be energy investment. Okay, ATP to ADP. Glucose will form fructose 1,6 bisphosphate, then glyceryl type 3 phosphate, then 1,3 bisphosphoglycerate, then finally pyruvate. So a lot of steps have been skipped over here, but the first step is the energy investment step, a phase, and the second phase is the energy payoff. So these are two phases of glycolysis. This phase, preparatory phase, and the payoff phase. So you can see in the preparatory phase is a phosphorylation of glucose and its conversion to glyceraldehyde 3-phosphate. For this, these are the structures. These are basically the products that are being formed. There's a first priming reaction which requires energy, ATP to ADP. Then second priming reaction which again requires energy, ATP to ADP. Then we have the cleavage of 6-carbon sugar phosphate to the 3-carbon sugar phosphate. And these are the enzymes that are being utilized in each of the steps. Okay. Then is the payoff phase, the oxidative conversion of glyceraldehyde 3-phosphate to pyruvate and the coupled formation of ADP and NADH. Okay, first oxidation and phosphorylation, then first ATP forming reaction, substrate level phosphorylation, then second ATP forming reaction, substrate level phosphorylation, and these are the different enzymes that are being used. So this is the preparatory phase. As I said, the different structures, the different products, and the different enzymes that are being used. So the reaction one is phosphorylation, where glucose gets converted to glucose 6-phosphate, with the utilization of energy, and magnesium ion is also required. This gives rise. This is done by hexokinase. So phosphorylation of glucose ensures that pathway intermediates remain in the cell. So glucose gets transformed to glucose 6-phosphate ATP to ADP. Phosphorylation occurs on C6 and C1 is a carbonyl group and cannot be phosphorylated. So hexokinase versus glucokinase is a study that have been done by people. Hexokinase 4 is also called glucokinase. So these are tissue specific isozymes. The next step will be the reaction 2, which is isomerization. Okay, so in this reaction, glucose 6-phosphate is converted to fructose 6-phosphate with the help of magnesium ions as well, and the enzyme is phosphohexose isomerase. Okay. So from the aldose, it is converted to a ketose group now. Okay. So the glucose 6-phosphate isomerization moves the carbonyl to C2, a prerequisite for 3 and 4, fructose 6-phosphate. Sorry. So the reaction 3 is then phosphorylation, where fructose 6-phosphate gets converted to fructose 1,6-bisphosphate, phosphofructokinase 1, ATP to ADP, magnesium 2 plus is the ion, divalent ion being used. Okay. Then this in now C1, now a hydroxyl group can be phosphorylated. This ensures that both products of CC bond cleavage are phosphorylated and thus interconvertible. Fructose 6-phosphate to fructose 1,6-bisphosphate. Then reaction 4 is the cleavage reaction. Fructose 1,6-bisphosphate with the help of aldolase gets converted to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Reaction 4 is cleavage, where the carbonyl group at C2 facilitates CC bond cleavage at the right location to yield 2,3 carbon products by the reverse of an aldol condensation. Fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate. It can also get converted to dihydroxyacetone phosphate. Reaction 5 is isomerization, where dihydroxyacetone phosphate gets converted to glyceraldehyde 3-phosphate, and this enzyme is triose phosphate isomerase. So this keeps on happening, reversible. Then reaction, so this is interconversion of the two products of this funnels both products into a single pathway. So we need to keep track of the carbons, okay? So derived from the glucose carbon 1, 2, 3 and derived from the glucose carbon 4, 5, 6. Okay, that's how we move ahead. There is a payoff phase where oxidative conversion of glyceraldehyde 3-phosphate to pyruvate and the coupled formation of ATP and NADH. Okay, so in the first oxidative oxidation and phosphorylation occurs. The enzyme is glyceraldehyde 3-phosphate dehydrogenase. Then next first ATP forming reaction substrate level phosphorylation occurs when 1,3-bisphosphoglycerate is converted to 2-3-phosphoglycerate and the enzyme is phosphoglycerate kinase. Then 3-phosphoglycerate is converted to 2-phosphoglycerate. Enzyme is phosphoglycerate mutase. Then 2-phosphoglycerate is converted to phosphoenol pyruvate. The enzyme is enolase. And then 2-phosphoenol pyruvate is then phosphoenol pyruvate gets converted to pyruvate, and that enzyme is pyruvate kinase. And the reaction is second ATP forming reaction, substrate level phosphorylation. Then reactions is oxidation. So glyceraldehyde 3-phosphate plus inorganic phosphate will give rise to 1,3-bisphosphoglycerate. Okay, so oxidative phosphorylation of glyceraldehyde 3-phosphate with 1 NADH produced is a prerequisite for ATP production. Reaction 7 is substrate level phosphorylation when 1,3-bisphosphoglycerate will give rise to 3-phosphoglycerate with magnesium ions and phosphoglycerate kinase. Now 3-phosphoglycerate and 2-phosphoglycerate can be interconverted using phosphoglycerate mutase. Now 3-phosphoglycerate to 2-phosphoglycerate, the remaining phosphoryl group moves from C2 to C3, setting up the final steps of the pathway. 3-phosphoglycerate, phosphoglycerate mutase, 2,3-bisphosphoglycerate, 2-phosphoglycerate. Reaction 9 is then dehydration. 2-phosphoglycerate will get converted to phosphoenol pyruvate using enolase. The phosphoenol pyruvate would get converted to pyruvate using pyruvate kinase, where magnesium and potassium are used. Okay, so these are the 10 steps involved in glycolysis. Now, all carbohydrates enter glycolysis in muscle often via hexokinase. So there are feeder pathways as well. So, what is the exact meaning of? One second, let me connect my charger. Okay, 1 second. So feeder pathway for glycolysis at least is something that basically feeds glucose or another starting material into the pathway. Okay, that's a feeder pathway for glycolysis. So if you talk about glycogen breakdown, okay, we know that glycogen phosphorylase catalyzes simultaneous phosphorylation and cleavage of an alpha 1,4 linked glucose from non-reducing end of glycogen, and this reaction is called phosphorylysis. Okay, so glycogen breakdown, if you're talking about this, the first step is glycogen phosphorylase. Okay, this is basically what happens in case of glycogen phosphorylase. We'll get glucose 1 phosphate, glycogen N-1 glucose units. And glycogen breakdown will be caused by phosphoglucomutase. So glucose 1 phosphate, glucose 1,6 bisphosphate, glucose 6 phosphate. So G6P fate depends on the tissue. In muscle, G6P pursues through glycolysis, and in liver, G6P is converted to glucose. So glycogen breakdown, we have glycogen that will get converted to glycogen phosphorylase, glucose 1 phosphate molecules. In the transferase activity of deep branching enzymes, so alpha 1,6 glucosidase activity of deep branching enzyme. Then you have the unbranched alpha 1,4 polymer substrate for further phosphorylase action. There are glycogen storage diseases. So what are the fate of the products pyruvate and NADH? Okay, so pyruvate can either get converted to ethanol and carbon dioxide, that is the fermentation to ethanol in yeast, hypoxic anaerobic conditions, or it can get converted to lactate fermentation to lactate in vigorously contracting muscle in erythrocytes, in some other cells and in some microorganisms like anaerobic conditions, or in under aerobic conditions it can give rise to 2-acetyl CoA with the release of 2 carbon dioxide or citric acid cycle, animal, plant, and many microbial cells under aerobic conditions. So this is a fermentation in animals. Lactic acid from skeletal muscle is sent into the bloodstream, and lactate threshold occurs when production exceeds clearance, so glycolysis cannot continue. Okay. So how can glycolysis be regulated? Glycolysis starts with the phosphorylation of glucose, the 6-carbon molecule, right? So the irreversible steps are regulated like hexokinase or glucokinase step, the first step, then the phosphofructokinase step, and the pyruvate kinase steps. So control of hexokinase. How is the step by hexokinase controlled? So glucose plus ATP will give rise to glucose 6 phosphate plus ADP, the feedback inhibition by glucose 6 phosphate. So when there is excessive production of glucose 6 phosphate, it will go and negatively inhibit hexokinase, like a negative feedback. Okay. By control of PFK1, how does that happen? So there is fructose 6 phosphate plus ATP will give rise to fructose 1,6 bisphosphate plus 8 ADP. So the citrate and fructose 2,6 bisphosphate ATP and AMP ADP AMP and ADP and fructose to 6-phosphate will increase the rate of the reaction, whereas ATP and citrate will probably inhibit. So ATP is an allosteric inhibitor of PFK1. That is how control of PFK1 happens. So two binding sites, substrate and allosteric site. So low ATP will give rise to good PFK activity, high ATP will give rise to low PFK activity because ATP is an allosteric inhibitor of PFK1. How does the control of pyruvate kinase happen? So pyruvate kinase will be formed. Pyruvate and ATP will be formed, and this process can be inhibited by acid alcohol long chain fatty acids. Okay, so in liver, glucagon will increase the rate of pKa. Okay, but in all other glycolytic tissues, acid alcohol long chain fatty acids will probably act as inhibitors. So what's the control of glycogen phosphorylase? Now phosphorylase B, which is the inactive form, gets inactivated by phosphorylation into phosphorylase A, which is an active form. This leads to glycogen breakdown. Now glycogen phosphorylase is activated upon phosphorylation by phosphorylase kinase. So phosphorylase kinase is activated upon phosphorylation by protein kinase A, and protein kinase A is activated by cyclic AMP, which is produced by a G protein in response to epinephrine glucagon. So these are the three steps. Okay. So phosphorylase B is a less active form. Phosphorylase B kinase converts phosphorylase B less active form to phosphorylase A active form. So what happens to the glucose when it gets oxidized via the pentose phosphate pathway? So the glucose 6-phosphate will get converted to 6-phosphogluconate by glutathione reductase, and then it will get converted to ribulose 5-phosphate and then to ribose 5-phosphate and etc. So ribose 5-phosphate is necessary in rapidly dividing cells. So transketolase requires thiamine pyrophosphate as a coenzyme. So these are the reactions. The reactions 1 and 3 will produce NADPH, and reaction 4 here will produce ribose 5-phosphate. And the key enzyme will be glucose 6-phosphate dehydrogenase. Now this is allosterically inhibited by NADPH and activated by NADP+. So that is the kind of regulation that happens in this case. In the oxidative phase, some cells need NADPH but not ribose 5-phosphate. So ribose 5-phosphate can be recycled in the non-oxidative phase. Then the pentose phosphate pathway is a non-oxidative phase, as you can see over here. This is the oxidative reactions of the pentose phosphate pathway. Okay, so that brings us to the end of the question that we started with. If we go back to the question, so the enzyme for phosphorylation of fructose 6-phosphate is called PFK2 to separate it from another enzyme PFK1 that catalyzes the formation of fructose 2,6-bisphosphate from fructose 6-phosphate in a separate pathway. And in some organisms, fructose 2,6-bisphosphate is a potent allosteric activator of PFK2. So that was all about phosphofructokinase and allosteric inhibitions. Okay. So we can stop our session here today. If you have any questions, you can ask me, or else, you can stop the session. Do you have any question? Okay, since if you don't have any question, we can stop the session for here today. All the slides will be uploaded and the videos will be uploaded on YouTube in the public domain. So we'll meet again next Monday, same time. Thank you so much.